I'm recording now.
Perfect.
Okay.
Thank you.
Do you hear me?
Yeah.
Okay. And you see the presentation. So you said you were going to present me to make a short introduction.
Mhm.
Bailey?
Yes. Yeah.
We are just a little slow to start Esperanza in that people in the department filter in until 10 minutes after 1, sometimes a little bit longer.
Okay, no problem. No problem, it's perfect.
I've lost my screen. Okay, there we go. All right. We can start in just a minute once I get my screens figured out.
Bueno. Bueno. Sí, la escucho. Y nadie le abre. Ah, es el 112, es a la derecha, es donde está el portón cerrado. Ajá. Ya sé, no puede ni siquiera entrar a la privada. Déjeme mandarles un correo para que le abran. Perdón. Un momentito, entonces me voy a desconectar de aquí para poder mandar el correo.
Okay, I think we can go ahead and get started, and then people will come in. So, hello everyone. Welcome to this week's Raymond seminar. Today we have Dr. Esperanza Martinez Romero from UNAM talking to us about endophytes, which is very exciting. So I'm going to take a minute to introduce Dr. Martinez Romero in a minute, but first just want to remind everyone to keep your mics muted during the presentation, and we'll have some time for Q&A at the end. Hi, Dan. So I'll go ahead and introduce Esperanza now. Professor Martinez Romero is a professor and head of the Genomic Ecology program at the Genomic Sciences Center at UNAM. From 2013 to 2019, she was coordinator of the bachelor's program in genomic science at UNAM, and she teaches undergraduate and graduate courses. She has been the main advisor of 56 theses, of which 26 were doctoral theses. Esperanza Martinez Romero studied biomedical research at the National University of Mexico for her bachelor's, master's, and doctoral degrees, and she was a postdoc at the National Institute of Agronomic Research in Toulouse, France. She has also done research stays in Sweden, Germany, Brazil, and sabbatical years at the University of California, Davis. She's reviewed many articles submitted for publication, and she is a member of various journal editorial committees and has been invited to present opening and closing lectures and keynote speeches at various national and international congresses. She was president of the Mexican Association of Microbiology and is a member of the American Academy of Microbiology and American Society of Microbiology. She has participated in juries for awards and evaluations of researchers from abroad. Dr. Martinez has obtained funds from CONACYT, PAPIT, and VLIR. FAO, and the Inter-American Institute for Cooperation on Agriculture. She studies the mutualistic symbiosis of bacteria with plants and animals native to Mexico using metagenomic and functional genomics approaches. She was a pioneer in the molecular study of the nitrogen-fixing symbiosis of beans and endophytes of corn and beans. She has given workshops and advice to agricultural producers, and she described new species of bacteria from plants and insects from Mexico. Some of them nitrogen-fixing bacteria, in fact. And the strains that she obtained have been deposited in the official bacterial collection, and some of them are used as inoculants or biofertilizers in agriculture, which is super cool. She's received awards and prizes such as the National University Award, the Mexican Academy of Sciences Award, and recently the National Science Award of Mexico in December of 2019, and the UNESCO Prize for Women in Science in March of 2020. She has organized national and international congresses. In 2020, she organized the National Microbiome Congress, and in 2021, the Latin American Microbiome Congress online. She's been recognized as a highly cited Mexican scientist and has published 216 articles in international journals, 23 book chapters, and 11 genome announcements. She's received more than 20,400 citations to her published work with an H index of 80. We're really excited to have Dr. Martinez Romero here talking to us. And without further ado, I will turn it over to her and let her give her presentation.
Thank you, Bailey. Thank you, Eric, for the invitation. I will tell you about different stories, and the first one starts with Phaseolus. Phaseolus was our first model. Phaseolus vulgaris, common bean, is widely consumed worldwide. And we decided to study the bacteria that form nodules in Phaseolus. Phaseolus is a very widespread genus in Mexico. And we are the country with the largest Phaseolus diversity. Why do we have that large diversity? It may be in relation to Mexican orography with lots of mountaintops, where mountaintops are being viewed in ecology as sky islands. So in different mountaintops, Phaseolus vulgaris diverge and are different from other mountaintops. This is interesting because when we sample them, we have to go to the mountains. And we are now limited because safety in those mountains in the Sierra is is not good now. There are safety issues now because lots of drug lords are now growing different type of crops that are illegal in that area. And so UNAM does not allow us to go sampling in those areas. But before we had all these problems with drug war and all this enlargement in drug production in the Sierra, we could go there and sample different Phaseolus in different sites of Mexico. So what is interesting for us is the nodules, is the nodule inhabitants. And therein we find different sort of genera of rhizobia that all have the capacity to form nodules. So we did some years ago a search of some Phaseolus species, and I must say this is very limited. This is like the peak of the iceberg of Phaseolus diversity nodule bacteria. And we sampled some many Phaseolus that were not domesticated. And we found that most of them nodulated form nodules with bradyrhizobium, while the most modern temperate species form nodules with rhizobium. Among the ones that were quite interesting was for us Phaseolus albescens. Phaseolus albescens is a vine that climbs over pine trees high in the mountaintops, and the plant may be over 18 meters long, climbing on the pine trees. we got seeds and we planted them in the lab, and we were very happy to get these long, long vines in some wood supports. And after a moment, they never prospered. They died. All the time they died. We couldn't have them growing in the lab. Phaseolus albescens is a relative of Phaseolus vulgaris, is a very close relative of Phaseolus vulgaris. But we were, even though we couldn't grow them in the lab, we obtained nodules from the field. And from nodules in the field, we isolated bacteria, and we found that they were quite related to those from Phaseolus vulgaris. We tried cross-inoculation experiments, and we found that those from albescens and vulgaris were not fully effective in heterologous hosts, meaning that there were some adaptations to their related hosts. So research on Phaseolus vulgaris is rather limited. It's limited because we don't have we had only like 6 seeds, and we are unable to recover more because of these unsafety issues. But with all the data from these and other Phaseolus species, we could identify that perhaps the oldest symbionts in Phaseolus were Bradyrhizobium, and not the Rhizobium species that we normally isolate from Phaseolus vulgaris. So we got an idea that perhaps there was a symbiont shift from Bradyrhizobium to Rhizobium in Phaseolus, and the most modern species form nodules with Rhizobium, while most of the plants in the Phaseolea form nodules with Bradyrhizobium. And this was related to some, a proposed transfer of genes from Bradyrhizobium to Rhizobium. So a phylogenetic study showed us that some of the genes that are required to render the bacteria more specific towards Phaseolus were perhaps acquired by lateral transfer from Bradyrhizobium. Bradyrhizobium has the largest diversity of these node decorating genes that add fucose to the node factor. And the genes from Bradyrhizobium adding fucose to node factor are quite diverse. And those from Rhizobium are just very limited lineages. That these bacteria were probably acquired, that Rhizobium probably acquired the genes from Bradyrhizobium. It's not my phone. Someone else's microphone is interfering. So we have this hypothesis that Rhizobium became more adequate by acquiring genes from Bradyrhizobium. What's going on?
Yolanda, can you mute?
Oh.
Sorry, Esperanza. Go ahead.
Okay. So, well, this was very interesting because we always consider that Rhizobium etli, Rhizobium fascioli, that are the canonical symbionts of Phaseolus vulgaris, were the best symbionts. But it turns out that maybe it was a symbiont shift that led us to work with fast-growing bacteria instead of slow-growing bacteria. In collaboration with a group in Peru, we have characterized bacteria from Phaseolus lunatus, and we also have some Phaseolus lunatus isolates from Mexico, and all those are Bradyrhizobium, these slow-growing bacteria. Bradyrhizobium is very interesting, though it is slow grower and very difficult to make genetics, because Bradyrhizobium seems to be the oldest genera that had the oldest genus that had not genes for fucose addition in the node factor are, I told you, are very diverse in bradyrhizobium, but also other node genes are quite diverse in bradyrhizobium. For example, node A, node C, that make the core of the nodulation factor. And they are also they also exhibit the largest gene diversity. So among rhizobia, I always have the hypothesis that bradyrhizobium is oldest. And now that we are looking at bacteria from trees, we see more or less the same story. Many groups of legume trees are nodulated solely by bradyrhizobium. In addition, bradyrhizobium forms nodules with with all the families of legumes and with Parasponia, that is a non-legume. So we have always announced that bradyrhizobium is like the oldest, but people say, no, don't be so happy, because maybe the node genes evolved in Actinobacteria, in Frankias, and not in bradyrhizobium. So maybe they were transferred from Actinobacterium to bradyrhizobium, and from brady to other rhizobial genera. So the origins of nodulation may be older and not even inside the alpha protobacterial group. Other people have argued that the origin of nodulation may be beta protobacterium, because they found some transporters that have homologous in the genome of beta protobacterium. So maybe there is no conflict. And some of the genes arose in some bacteria, and other genes that also participate in nodulation secretion, for example, maybe emerge in other bacteria. So there may be not a single origin of the whole cluster of nodulation genes. So I was telling you that we explored Phaseolus, and we wanted to explore the whole 50 Phaseolus species, and we have like 5 or 7 Phaseolus species that we have really analyzed. And those that were analyzed were mainly non-domesticated. And we believe that domestication didn't have an impact in the choice of Phaseolus symbionts. We wrote a short review on plant microbiota modified by plant domestication. And here we discussed that that plant microbiota needs not to change with domestication if the traits being selected by domestication are not affecting the symbiosis relationship. And so in many cases, domestication does not need to affect the symbionts. So we made sort of a little cartoon showing that while plants may be very, very diverse, and the domestication effect makes like a bottleneck, and finally you finish with some varieties that are suitable for human growth, for human cultivation. And what happens with the microbiota? The microbiota associated with wild type plants may be very large. And when you make the domestication process, you may select different populations of bacteria that form nodules. But we discussed that in a case that forms associations. But in the case of nodules, maybe preserving the ancestral symbionts gives an advantage. So Phaseolus vulgaris domestication, it is very clear that led to a diversity of phenotypes, because people select colors of seeds because maybe they are related to taste. And so in the Mexican traditional agriculture, women are so proud to carry big baskets full of seeds of all colors of Phaseolus vulgaris. So they really select diversity. And in a certain way, this leads to another spread of Phaseolus in different places. So the bottleneck that we saw before, where we lose diversity, is rapidly changed, is rapidly compensated by diversification processes carried by humans that grow beans in many different gradients of altitude, temperatures, humidity. So we are always selecting for diversity inside domesticated plants. So there's like a burst of diversification, and the different Phaseolus also are very promiscuous. The domesticated Phaseolus are very promiscuous and form nodules with many different bacteria that they encounter, maybe in their different places where agriculture is maintained. So we really found in Phaseolus that domestication did not affect rhizobial preferences, because we compared wild Phaseolus vulgaris and domesticated ones, and we saw no differences in their symbionts. I was telling you that Bradyrhizobium is a very interesting genus because it is like the original one, and it has symbiosis islands instead of plasmids. And the symbiosis islands, in principle, are not easily mobilized in the laboratory. But in nature, it seems that they move. Because when we make phylogenes comparing Bradyrhizobium housekeeping genes with those from symbiotic islands, all in the chromosome, we see incongruences, evidencing that there is some sort of flow of genes involved in symbiosis. So the symbiosis islands in Bradyrhizobium are very interesting, involved in some way the plasmids, the plasmid organization and the plasmid content that exists in a rhizobium. And we have characterized the symbiosis islands from some of the Bradyrhizobium from Phaseolus lunatus, lima bean, from Peru, and nowadays we are characterizing them from legume trees. So this I discussed. Bradyrhizobium may be the ancestral symbiont in Phaseolus, but also in legume trees. Bradyrhizobium is the only, I'm repeating, the only genus that forms nodules in non-legume families, and in the non-legume Parasponia, Bradyrhizobium genes are the most diverse. And the trees are very interesting because they have a large capacity to fix nitrogen. So nowadays, we are working with bacteria from legume trees, for example, from these Lysilomas or from Ingavera from Brazil. And there, the effects of nodulation are very evident. Even in soil, if you don't add the inoculant, the Bradyrhizobium, the development of the plant is very, very little. The plant grows scarcely. And when you add the Bradyrhizobium, the effects are notorious in these plants. And this may be seen also in Leucaena. But Leucaena forms nodules also with Rhizobium tropici, with Rhizobium grahami, with other Rhizobium species, and not with Bradyrhizobium. And we believe that this is a further acquisition in legume trees, the nodulation with Rhizobium. It's a similar story to what we saw in Phaseolus, and we have not published on this yet. So the mobile elements in nodule plants led us to the concept of symbiobars. Symbiobars are refer to the host specificity, and in Rhizobium and in some in some of them, in some Mesorhizobium and in Cynorhizobium, the specificity towards the plant is encoded in plasmids, while it is encoded in symbiosis islands. And this leads to a large flexibility in the possibilities of nodulating from a single species. The largest number of symbiobars is found in Smellilotti, and it may have capacity to form nodules in Acacia, in Medicago, in other legumes, depending on the symbiosis plasmid that it harbors. In contrast, we may find that a single species may have, well, in this case, it's a single species that may have different symbiosis plasmids. So there are different symbiobars in a single species. In other cases, a single symbiobar may be found in different bacteria. For example, Rhizobium etli has symbiobar fascioli, Rhizobium fascioli has symbiobar fascioli, Leguminosarum has symbiobar fascioli, Giardinia may have the same, and it is depicted here as the orange plasmid that is contained in different bacteria. So this concept exists not only for Rhizobium, but also for Bradyrhizobium with their symbiosis islands in the chromosome. So genomic islands have adaptative genes that are useful for nodulation of some plants. In general, it was thought that Bradyrhizobium were all generalists and that tropical legume plants were not really very selective for nodulation. And that gave an advantage because in tropical forest, density of legume trees is low, is rather low. You don't have a real forest, like the pine forest, of a single legume. You have one legume here and another one there. And why is this so? Maybe it's because the pressures of pathogens, of fungal pathogens, is very large. They are very aggressive in the rainforest. Fungi thrive in wet, in humid and high temperatures. And they love there. So it's a paradise for fungi. And then plants have to protect themselves, perhaps by having very low densities. And if they have low densities, then maybe they are symbionts. If they have very specific symbionts, it's very difficult to keep them with them. So it has an advantage if you have a very generalist symbionts that you may encounter everywhere. And that may be the case, but with Inga vera from the tropical legumes from Veracruz, we found that something that was counterintuitive, that the bacteria seem to have some specificity. They like their bacteria, but they are very diverse, the bacteria that can form nodules with Inga, meaning that perhaps there are many bacteria that have this capacity. But if we take those plants from Veracruz and take them to the center of Mexico, Veracruz is near the Gulf of Mexico. And it has the northernmost rainforest in North America. And if we take them to the center of the country where the climate is completely different, they don't form nodules there. And then they are limited for nitrogen fixation. So there seems to be some sort of specificity, and we are trying to explore the genomic islands that make different symbiosis in these trees. So besides having a rich fasciolus, having a richness of fasciolus species, we also have a richness of Fasciolus vulgaris associated plants, because in Mesoamerica, agriculture involved not only the legume, but a cereal. And the way bean was grown was in combination with maize. Maize and bean have been partners for around 7,000 years. And that was the way the traditional agriculture was performed in such a way that in a single plot of land, you could grow a cereal, a legume, and also, for example, cucurbitáceas, that are the zucchini, these that we have also a large diversity. So there may be other plants associated with the milpa, including bananas. So that's the way it used to be. But modern agriculture decided that this was not very practical, and so they separated bean and maize and made the monocultures and added lots of fertilizers to both maize and Phaseolus vulgaris, and counter-selected Phaseolus vulgaris symbiotic capabilities. So finally, they ended with very small bushes of beans that were easy to harvest, but they didn't have a good modulating capability. So to look for milpas now, it's a big problem because it's not the way the modern agriculture is performed. And why we were interested in milpas, because there maize and bean share their roots, share the place, and the roots are very close one to each other, so they could share their endophytes. They could share the bacteria. And Eureka, we found the bacteria from bean inside maize plants. And they are endophytes, natural endophytes. And they are very helpful to inhibit fungal pathogens in maize. Rhizobia really counteract the fungal pathogens in maize that are very frequently found otherwise. And very limited levels of nitrogen fixation may be attained with rhizobium in maize. In an effort to have nitrogen fixation in maize, we designed an experiment where we added several nitrogen fixing bacteria to maize roots in controlled conditions in the laboratory. And we found expression of NIF genes from Rhizobium fascioli, but we didn't find nitrogen fixation measured by acetylene reduction in maize plants. However, we found nitrogen fixation by azospirillum, and the effects of maize inoculation of azospirillum was very clear. And we published this last year in the metatranscriptomic analysis on maize rootlets. And I think it's a very nice example of evaluating the effects of artificial bacterial communities in a cereal. More recently, this year we just published a paper on only Rhizobium fascioli in milpa, in the laboratory. We have both maize and bean in flasks, and we made a transcriptomic study of Rhizobium fascioli as a single plant. And therein we found effects of inoculation, clear effects of inoculation. It's a very recent paper in Frontiers in Microbiology, and the first author is Aguirre Noyola, and he just got his PhD yesterday. And we were very happy and busy with his exam yesterday, and he got this special mention because the work was very good and the presentation was great. And so there is hope for Rhizobium in milpas, for Rhizobium from beans in maize. When working with plants for a long time, we decided to expand our scope, and we started working with gut bacteria, especially from insects native to Mexico. And we found that bacteria in guts of insects that feed solely on plants resemble those from plants. And we found lots of similarities of gut and root bacteria. And among the similarities, there could be nitrogen fixation. Nitrogen fixation may occur in guts, and termites have been known to form symbiosis with nitrogen-fixing bacteria from 1973. Two papers were published, one in Science and another in Nature, on nitrogen fixation in termites. So we said, there must be other insects that fix nitrogen. And the same question was asked by Jurkevich in Israel, and he worked with fruit flies. And he found nitrogen-fixing bacteria in fruit fly guts. So these are the termites. Termites are very well known for their capacity to feed on wood, and they are terrible pests for humans. And they have lots of nitrogen-fixing bacteria in their guts. And so we went to the zoo, and we found that this nice animal and some tortoises had nitrogen fixation in their feces. And nitrogen fixation was measured in the feces there in the zoo from an African tortoise, and there we changed gears and went to a Mexican tortoise in the north of Mexico, that is Copherus berlandieri. And we found nitrogen fixation in all the samples of Copherus berlandieri feces. And we isolated the nitrogen fixing bacteria, just like we do with endophytes from plants. That is in small tubes where we have a gradient of oxygen because we have very soft agar and we have no nitrogen. And there we make serial passages in those conditions with no nitrogen that would allow nitrogen fixers to be enriched. And we isolated Klebsiella varicola. Klebsiella varicola was well known to us because we named it. We named it some years ago as a plant-associated bacterium, but also as clinical isolates. Because when we were working with these plant-associated isolates, a colleague from us from the Health Institute told us that he had a collection of Klebsiellas from little children in pediatric hospitals in Mexico. And he wanted to compare them to our plant isolates. So we got his collection, and around 10% of the isolates from humans, from little children, corresponded to Klebsiella varicola. They were undistinguishable by the approach we did then, sequencing several marker genes. And finally, now the genomes from many Klebsiella varicolas show that indeed Klebsiella varicola is a plant endophyte, but also is a clinical bacterium. And the differences between plant and clinical isolates is mainly the levels of nitrogen, of antibiotic resistant genes, and also of virulence genes. Those from plants have normally no virulence genes, and those from the tortoises, they do not have, they are like the ones from the plants. They do not have high levels of antibiotic resistance. They have barely only one gene for ampicillin resistant, that is chromosomal born in Klebsiella, and all of the Klebsiellas have this gene, and but they don't have the plasmidic genes for many antibiotic resistances. So what? We found Klebsiella varicola in tortoise feces. And also something that was very peculiar of the isolates from tortoise feces was their genetic similarity. Even if they were from different tortoises, they shared a very a common pattern of ERIC PCR profiles that indicated that they were not highly diverse, that they were limited clones that were maintained within the tortoises. So how is this done? This may be because tortoises like to eat feces, and when they are born, they eat the feces from their mother. So in a certain way, they perpetuate clones of Klebsiella in feces. That case of passing bacteria from mother to offspring is seen in other animals, like koalas. Koalas pass their bacteria from mother to offspring because those bacteria are very precious, are the best a mother can give to their children. They allow the baby koalas to eat the eucalyptus leaves that are very hard to digest. So if they eat the mother feces, baby koalas acquire bacteria to digest the fibers of plants that are very difficult to digest. So we have this collection of Klebsiella varicola from tortoises, and we want to know if they can promote nitrogen fixation. And we are trying now different model animals like zebrafish or chicken to see if we can get evidence of nitrogen fixation in animals. We just started a first experiment with chicken that we saw some differences, but not with Klebsiella varicola. We saw some difference with Azospirillum as an inoculant. We want to repeat all the experiments again to be able to put in evidence nitrogen fixation in animals, and that's something we will pursue in the near future. So Klebsiella varicola from herbivores, tortoise feces, not turtles, tortoise. We found that they were not virulent in mice. They were not virulent in zebrafish. They were not virulent in galerias. Galerias are these very small insects that are now being used in pathogenicity studies. Interestingly, we found that it excretes alanine when it is fixing nitrogen. And that reminds us of a report in Bradyrhizobium where they said that alanine was also excreted and not ammonium in Bradyrhizobium under some conditions. It is very interesting because alanine would not be toxic to mammals, while ammonium is toxic to animals. So if we want to export nitrogen fixation to animals, we would have to have something like this nice Klebsiella varicola from tortoise feces. It was a pity that we didn't find that Klebsiella varicola in chicken promoted growth, but we will try other lines of chicken because maybe we didn't use the most adequate one because it was like an obesity model for chicken because it's used to be fat. So it was not the best experiment we made, but we will try it again. So in the meantime, we have all the genomes from Klebsiella varicola, and we have, as I told you, the genome from varicola from tortoises didn't have genes for antibiotic resistance as clinical ones, and did not have genes for virulence. So with all this story, we started thinking that there was perhaps a more global flow of bacteria between plants and animals. So we thought of a plant and animal microbiome integration, and that's why I put this title in the seminar. We and herbivores eat endophytes. So we know that all plants have endophytes, and every animal that eats plants finally eats the bacteria that are inside the plants. And why do we emphasize endophytes? Because endophytes, by being inside plant tissues, have sort of an enteric capsule that protects the bacteria from digestion, from the chlorhydric in the gut, in the stomach, and from the biliary salts in the early small intestine. And finally, these bacteria can find all their way to the gut, to the colon, where the fiber plants are digested and the bacteria are liberated. So a source of gut bacteria for animals is plants. And if we eat plants, we eat the bacteria that are in the plants. And if we eat them, we excrete them. We may excrete them and they finish in the feces of the animals. And from the feces, we can recover them. Just to remind you that all the human gut microbiome has been made mainly with feces and not really cutting the gut of humans. So we proposed a cycle, an endophytic enteric cycle, where the feces may become the inocula for plants. And finally, there is a population of plant and gut bacteria that resembles. So Wasserman published, a group in Austria published that we eat 100 million bacteria with an apple. And that is the case with most raw legumes that we eat. Even if a lettuce, a banana, or anything is disinfected, washed, or peeled, we still eat endophytes all the time. So, and it is clear that herbivorous animals have the most diverse gut microbiome, and that was a paper by Ley in Science 208 in 2008, where he said gut of herbivorous animals is very diverse, is more diverse than that of any carnivorous or omnivorous animal. So if we eat endophytes, we can find them in the digestive tract. And I told you about our story with tortoises, but we have also explored microbiome of butterflies that also feed on plants, and of these wonderful cochineals, carmine cochineals, that feed on cacti and produce a carmine color. We also explore those from other worms from the agave that is used to produce tequila. And I was telling you about the story of the koalas before. Koalas are well studied because of their capacity to feed on lots of polyphenols. And with all that information, we captured and made sort of a study and said, well, which are in plants and which endophytes we recognize from the guts of koalas, pandas, rabbits, tortoises, and many of the bacteria that are reported as gut bacteria are clear plant endophytes. So that's a source of gut bacteria. But attention, please, because here I told you that the plant fragments may be well maintained and maybe are not released, all of them in the colon. So in the very same paper, we made a call of attention saying, look, we are getting gut bacteria in all these thousands of microbiome studies. And maybe some of them are not bona fide gut bacteria. They are still in the fragments of fibers in the colon. So maybe they are not delivered, and they are still plant bacteria and not gut bacteria. So be careful. And that happened to us because when we were isolating the feces to make a metagenome, when we were extracting DNA from tortoise feces to make the metagenome, my student came and said, this is horrible. This is real horrible because it's only fibers, plant fibers in the kisses of the tortoise. And the fibers contain bacteria. And that doesn't mean they are gut bacteria. So I think there are many mistakes in the microbiome studies that erroneously consider bacteria in plants, in plant fragments, as bona fide gut bacteria. So finally, well, this is a cycle that I just showed you. And when making a phylogenesis of the bacteria from plant or from the gut, what we see is that they are intermingled. And that's a prediction. If you are having a single pool of bacteria that circulate among plants and guts, you will not have clades that speciate, but rather intermingled phylogenies that mean that these bacteria go from one pool to the other, that circulate normally, and that there is a flow. And that we found for some of the well-known gut or plant bacteria like Clostridium, Lactobacillus, or Weissella. So finally, I want to thank UNAM and my institution, the Genomic Science Center. I want to thank my techniques, Toño, Mónica, and Julio, my colleagues, and support from grants from UNAM, PAPIT, and from CONACYT. Thank you very much for your attention.
Thank you so much, Esperanza. It was great and very, very interesting. I learned a lot. I just wanted to ask if anyone has any questions they would like to ask at this time. I definitely have one, but
Okay.
I wanted to like let other people chime in first.
Okay.
So I'll go ahead and ask mine, I guess. So my question is, like, what, given that plant endophytes might impact gut microbiomes and thus human health, what application do you see for these studies and this link between the microbes we find in agricultural systems and
Yes.
human health? Yeah.
That's a great question. And indeed, some people have started to ask, how can we manipulate plant bacteria, especially endophytes, to make them suitable for human health? So a researcher in Israel made a patent to try to put probiotics inside plants, probiotics that would help humans inside plants. In another case, there's a Mexican CEO that works in a company in the States. He is isolated from plants bacteria that could help human health in such a way that, for example, he has already isolated bacteria that could help women avoid the loss of bone as women age. He has a consortium of 3 bacteria, among them Lactobacillus plantarum, that could help inhibit osteoporosis. And I read, like in the news, in the media, that they are trying to put some compounds in lettuce that would help astronauts not to lose bones as well when they go in spatial missions. So I think there is large possibilities to survey bacteria associated to plants and not to have pathogens. Salmonella is a very good plant endophyte. And when it gets to plants, it contaminates them and it becomes an endophyte. And then later you can have infections with salmonella by eating plants. For example, recently also in the media, there was news in the States that people got sick from salmonella eating onions, yellow onions that came from Mexico. They were Mexican onions that were contaminated with salmonella. So which endophyte would you like to eat? Certainly not salmonella. And certainly some good Lactobacillus plantarum. But how can you control that? We need to advance in that path to have a control of our plant endophytes in order that we eat favorable endophytes. So that's really, there's really lots of opportunities for research and development on that field, because it enhances the role of bacterium in plants, not only towards plant growth, to have good promoters of growth or good competitors of pathogens, but also to have bacteria that are beneficial for human health when we eat them.
I'll jump in next if there aren't other questions. So Esperanza, an enormous thank you to you. That was, as always, wonderful. There are so many questions. One of the ones that I'm still just curious about is, what do you think are some of the differences that make Rhizobia a temperate group? And I'm also curious about the biogeography of it. Do Rhizobia predominate instead of Brady at higher elevations, so that you see them at mountaintops? And yes. If so, for tropical highland plants, like in the highlands of Ethiopia, you see species there that are in the same genus as species from temperate Europe. And we don't really have a great explanation for how they ended up in the Ethiopian mountains and in Europe and not in between. Do you have a sense of how, but in Latin America, there are oaks in the highlands of some of the mountains, and then oaks are common in the Northern Hemisphere. If Rhizobia is temperate as a group, how did it migrate?
Yeah, yes. I think I don't have an explanation why Phaseolus shift hosts from Bradyrhizobium to Rhizobium. One of the possibilities that I also considered is that Rhizobium has genes in plasmids, nodulation genes in plasmids, and that really helps spread. So when legumes started spreading, because there's a flow, a biogeography flow of legumes from tropics to temperate regions. And that is, I think there is solid evidence that it happened like that, from tropics to temperate. And also when this flow occurred, maybe there was an explosion of a diversity of legumes. How to cope with them? Maybe it was easier to exchange plasmids than to exchange symbiosis islands. And then it brings them the explanation more to a genetic field than to an ecology field. Because I see that because I studied somehow the resistance of antibiotics in Klebsiella. And all Klebsiellas have this like mother genus of ampicillin resistant in the chromosomes. And that's like the ancient one, the one we found in plants, in plant isolates and in tortoise isolates. And the clinical ones, what do they have? They have plasmids. They have plasmids, and those are the recent ones. And plasmids are easy to transfer at high frequency, to transfer all this information. So maybe that's a key, the possibilities to generate diversity and to spread the nodulation phenotype in Rhizobium.
That's so cool. Fascinating. Yeah, that makes sense. I'm afraid I have to jump off myself in a minute or two. So thank you again. I'm like you giving a virtual seminar in 5 minutes. So I should make sure that I'm ready. Yes, thank you again.
Thank you, and very nice to see you, Eric, again, and to have the opportunity to talk to you. I would be glad to send papers, any you ask for, and thanks so much.
Yolanda, do you have a question?
Yeah, I was in transition actually during your talk. I had to come home, so I'm not quite sure if I completely got something, but I got the impression you were talking about different Rhizobium species and how you were talking about kind of an environmental influence on Phaseolus associations, and I was wondering if that's based on the occurrence of Rhizobium, or is it the plant, or is it a combination of those interactions that seem to drive these patterns of association?
Yeah, rhizobial association in legumes are well studied, have been studied for many years. Indeed, Rhizobium as an inoculant has been used for 120 years around. So the association is very successful because bacteria in the nodules are specialized to fix nitrogen that provide it directly to the plant. So there's not these nitrogen losses as when you add fertilizers. And plants may benefit largely from their associated bacterial nodules. Indeed, there are bacteria that are not so efficient, but I have told you the explanation that I think that it is our fault that some Phaseolus vulgaris are so bad to fix nitrogen, because they were selected under conditions that nitrogen fixation would not have a clear effects, because they add fertilizers, chemical nitrogen fertilizers. And in that case, you don't really need a fixing, an efficient fixing in Phaseolus, an efficient fixation in Phaseolus. But if you work with trees that have not been domesticated by human beings, then nitrogen fixation is very efficient. And trees really have a large biomass, so they really need to fix lots of nitrogen. So they are very, very good models to study efficient nitrogen fixation and some sort of specificity that may occur.
Thank you.
You're welcome.
So I think we'll go ahead and end the questions there. I just want to say thank you so much for rescheduling after last semester. And it was really great hearing from you.
Thank you. Nice to see you. Bye.
Nice to see you too. Have a good day. Bye.
Have a good day.